Starting with microscopic cells and ending with massive, real-world agricultural impacts, we're breaking down plant tissue culture. It's kind of this invisible science, right? But it's completely revolutionized how we propagate, protect, and really improve the food and flora that sustain us all.
So think about this. What if you could grow an entire forest from just a single tiny leaf? I know, it sounds like pure magic, or maybe something straight out of a sci-fi movie. But the seemingly magical concept, it's an everyday reality in modern biotechnology. And as we'll see in this explainer, you'll understand exactly how scientists are actually pulling off this remarkable feat.
Okay, here's our quick roadmap for the explainer. First, we'll look at the biological superpower of totipotency. Then we'll step inside the sterile lab. Third, directing growth with hormones. Fourth, we'll run through a real-world case study on the rare Micropropagation plant. And finally, we'll zoom out to look at the massive global conservation impacts.
Starting off with Section 1, the power of totipotency.
Now, the foundation of literally everything we're discussing today rests on this one biological phenomenon, totipotency. So what is it? Well, it's the ability of a single plant cell to divide and regenerate into an entire, fully functioning plant. You see, unlike most human cells, almost any plant cell, whether it's from a leaf, a stem, or a root, acts like a master blueprint. It holds all this raw, locked-away potential to just become a brand new, whole plant. Honestly, all it needs is the right environment to wake that potential up.
The roots of this idea go way back. In 1902, German botanist Gottlieb Haberland first proposed the concept. And, while his early experiments largely failed, he really laid the essential groundwork. Fast forward to the 1930s, an American physiologist, Philip White, actually managed to cultivate tomato roots in a completely sterile environment. But the really massive, field-defining breakthrough, that didn't happen until the 1950s, when Toshio Morishige and Falk Skog developed the MS medium. It's this standardized, heavily researched nutrient soup that actually remains the absolute cornerstone of tissue culture today.
Moving on to Section 2, inside the sterile lab. So living plants out in the wild are naturally covered in this microscopic jungle of fungi and bacteria. If you were to just drop an unwashed leaf into some nutrient-rich agar jelly, those microbes would immediately take over, gorge on the nutrients, and just completely kill the plant tissue. Because of this, the plant material has to be flawlessly sterile. Scientists use these really highly specific, intense chemical recipes. We're talking things like a 30-minute soak in calcium hypochlorite or a really quick ethyl alcohol wash. This rigorously scrubs the tissue clean without actually killing the totipotent cells inside. It's essentially the ultimate extreme spot treatment for plants.
And this process gets incredibly delicate, especially when we're dealing with orchids. Get this, a single orchid capsule can contain up to 3 million microscopic seeds, and they have absolutely no food reserves of their own. So to sterilize them, lab technicians have to follow this incredibly strict 4-step procedure. They literally soak the capsule in 100% bleach, dip it in 95% alcohol, and then set it on fire with a flame. I mean, it's pretty intense. And only after that dramatic purification do they aseptically scrape those tiny seeds onto a culture medium.
All right, Section 3, directing growth with hormones.
Okay, so once we've got these sterile cells sitting happily on their nutrient medium, we need a way to tell them what to become. Think of them as molecular steering wheels. We use two main plant growth regulators, auxins and cytokinins. If you supply an excess of auxins, you force massive root proliferation. If you use an excess of cytokinins, you trigger multiple shoots and stems to grow out. But here's the cool part. If you balance them equally, the cells just multiply into this raw, unorganized blob of tissue called a callus. Tweaking this precise chemical ratio is literally how scientists grow distinct plant organs on demand.
Let's look at Section 4, Case Study on Mucuna pruriens.
Mucuna pruriens, which is commonly known as alkushi in Bangladesh, is a highly valuable medicinal plant. People use its pods to expel parasites, and the roots actually act as a really powerful diuretic. But because of unregulated harvesting and severe natural habitat destruction, it has become incredibly rare. And this creates a really urgent, real-world stake for us. Without a laboratory micropropagation protocol to rapidly clone and save this specific species, we risk losing its medical benefits entirely.
To tackle this, researchers started by taking tiny leaf segments from the plant and placing them directly on an agar medium. Now, what you're seeing here, that green, friable clump resulting from the process, that is the callus tissue we just talked about. It's the direct result of a perfectly balanced media. I know, it doesn't look anything like a plant yet,
but every single cell in that undifferentiated clump is a blank slate. It's primed and completely ready to be directed.
Next comes the incredible journey of differentiation. By carefully tweaking those hormones, researchers force those unorganized segments to erupt into multiple leafy shoot buds. Then, once those shoots grew just a few centimeters, they were carefully separated and moved over to a completely different medium. This new one is a half-strength MS solution that's been fortified with auxins, and it's designed specifically to trigger that critical root development phase.
And finally, the payoff. We get a fully acclimatized plant. But there's a catch. You can't just take a super fragile plant from a humid, perfectly sterile test tube and shove it into harsh, natural soil. It would immediately go into shock and die. It's kind of like moving a pampered indoor pet out into the wild, you know? It requires a really careful transition. Scientists slowly open the culture vessels over several days just to harden the plant, before finally washing it and establishing it in a standard soil and compost mixture. And there you have it, a complete, healthy plant regenerated entirely from a microscopic fragment.
Which brings us to our final section, cultivation and global conservation.
The sheer scope of applications for tissue culture is staggering, mostly because it completely bypasses traditional agricultural limits, like shifting seasons or really slow growth rates. With this tech, we can rapidly conserve endangered species before they disappear forever. We can produce entirely disease-free crops for global farming. We can even grow vats of plant cells in massive liquid bioreactors just to harvest valuable compounds like caffeine, nicotine, or essential medicines, without ever needing to actually grow the full plant. We can even use somatic embryogenesis to mass-produce artificial seeds. It's wild.
But it gets even better. Sometimes this process induces genetic mutations. They're called sumoclonal variations, which actually allow us to actively upgrade plants. Through this exact method, scientists developed a sugarcane variant called Ono that is completely resistant to Fiji disease. They've cultured a citronella variant that yields 37% more essential oil. Think about that boost in production. There's even a banana variant that was specifically mutated to withstand heavy, crop-destroying rains. We aren't just saving plants, we're actively improving them.
So this brings us to a critical final thought. As climate change increasingly threatens our global ecosystems with extreme weather and completely unpredictable shifting seasons, traditional conservation might simply be too slow. Could tissue culture be the ultimate key to saving our most vulnerable plant species? By unlocking the totipotent power of a single cell, we might just hold the exact tools we need to rebuild our planet's forests, literally one sterile test tube at a time. Thank you so much for joining me on this explainer, and